Hello. For the families here present, I'm Stefano Berto. I'm assistant professor at the Medical University of South Carolina in the Department of Neuroscience. And my lab is basically dedicated to CHAMP1. So we have multiple grants and so on, but we study really CHAMP1 in our lab. So my lab has actually two different aspects. One is the basic neuroscience. We want to understand how CHAMP1 affects neurodevelopment, and a translational medicine side, where we want to actually develop therapeutics for CHAMP1 syndrome. For the basic neuroscience, this is like actually complementary because we use the same techniques also for the translational part. We use patients-derived in vitro models, iPSC lines, and we basically push them toward the neuronal development. And we use electrophysiological neuronal connectivity approaches such as whole patch clamp and MEA. And we also use genomics to understand actually whether gene expression is actually affected by CHAMP1 loss of function. For the translational medicine, we also we collaborate with the Cohen's lab at the MUC, and we develop RNA-based therapy for CHAMP1 syndrome. And this part is gonna made by Zach, my PhD student. So we know this, right? CHAMP1 is a high-risk developmental disorder gene. We know the function. We have 200 more or less confirmed mutations, I guess, like probably even more. It's a really severe, it's a high-risk gene. And we know that patients with CHAMP1 disorder have intellectual disability, Hawkins-like behavior, microcephaly, hypothonia, and so on and so forth. We have multiple mutations. The majority of them are a nonsense mutation, a frame shifting mutation. These are causing truncated protein. But we have also missense mutation as well. We know actually the function of CHAMP1 in mitosis. You heard that from Kozo Tanaka, amazing, with this amazing talk. We know that if we knock down CHAMP1, we affect actually the chromosomal segregation, and therefore mitosis is affected. And we know that CHAMP1 regulates kinetochore microtubule attachment and also is involved in DNA repair mechanisms during this process in mitosis. What we know about the neurons and what we know about brain is not so much. The first paper was again from Kozo Tanaka in 2022, and you saw this already in his presentation. He actually used the CHAMP1 loss of function mice, heterozygous mice, and what we found is that CHAMP1 seems to disrupt the spatial organization of the neurons and neuroprogenitors. There is a delay in neuronal differentiation, and also the mouse shows some behavioral deficits like anxiety-like behaviors and so on. But given the established role in CHAMP1 in regulating mitosis, we don't really know what it does in post-mitotic neurons, right? And this is actually like what we wanted to focus in our lab. So we took, as I mentioned before, we use iPSC lines. These like typical control lines, and we call it control 1. We have multiple control lines, but this like presentation is showing like the data from this control line. And this is like, the schematic of the protein of CHAMP1, right? So we have zinc fingers motif at the end, and some motifs in the middle. And we know that these, for instance, these are important for POC-zeta. This is important for binding for Rev7, this is HP1 alpha, and the SNP, and so on. And we have two actually mutation, two iPSC lines carrying two different mutations. One is a frame shifting mutation. Let's call it CHAMP1B for the sake of time. And the other one is actually the arginine 497, the CHAMP1A. And this is actually what happens. So, and we have two different clinical presentation where the CHAMP1B actually has a mild high intellectual disability, mild motor delay, and mild ASD-like behavior. The arginine 497 has a severe intellectual disability, severe motor delay, and ASD. And what we do is taking those iPSC lines, reprogramming those in neuroprogenitors, and push them toward the glutamatergic neurons. And we collect data at the in vitro 30 and the in vitro 60, more like immaturish neurons, and more maturish neurons at the in vitro 60. So why this is important? Because iPSC-derived neurons retain the genetic background of the donor, develop neuronal structures such as dendrites and axons as a living brain, sends and receive electrical signals, and also form complex networks. This is a representative image of neuroprogenitors stain for PAC6 and nestin. And here, like neurons at the in vitro 30, you see that they are basically branching out a little bit, but they are not that complex. And at the in vitro 60, they start to create like more complex and more dendrites and so on and so forth. So they basically mature more and they branch out more. These are like another, and the CHAMP1 actually is expressed in our models. So of course, in iPSC lines, in neuroprogenitors, CHAMP1 is in red. So we know that CHAMP1 is expressed there. And now the main question, right? Do CHAMP1 mutation change the function, structure, and network connectivity of developing neurons in vitro? So to tackle this question, actually, we have like 4 approaches. One is a neuronal activity and function. We use a patch clamp. And we record the in vitro 30 and the in vitro 60. We have a neuronal structure, and we basically, we use a 3D reconstruction and AV sparse labeling. We have network connectivity using multi-electrode arrays, and also we use genomics to understand whether gene expression is affected and kind of correlate with the electrophysiological deficits that we see in these neurons. So let's start with the neuronal activity and function. So this is a canonical action potential from a control lines. This is a very, this is the standard electrophysiology that you can, action potential that you have in a control line. And so basically what it happens is that like there is action potential, depolarizing, repolarizing, and so on. This is electrical activity across time. And this is what you should expect actually from, as I said, from a neurotypical line. This is actually CHAMP1A and CHAMP1B. As you can see, there is like a huge deficit in the electrophysiological property of these, on these lines. If we quantify the frequency of the action potential in the in vitro 30, there is like a severe deficit here in these 2 lines compared with the neurotypical line. However, this frequency actually reduces in the in vitro 60 and 70. So the CHAMP1 mutation reduces excitability of neurons, especially at the in vitro 30, but basically they pick up more or less as the same by the in vitro 60 and 70. But what was interesting for us is that when we classify actually the neurons by multiple action potential, single action potential, failure, or none, what we see is that this is the neurotypical, as I mentioned before. In the in vitro 30 and 60, we have more or less 80%, 90% of the neurons showing multiple action potential, while the in vitro 30 and 60 for CHAMP1A, there is a strong reduction, and the in vitro 30 and 60 for the CHAMP1B actually kind of pick up at the in vitro 60. So CHAMP1 mutations reduce actually the percentage of mature neurons that fire multiple action potentials. And what we know is also that neurons grow, both in size during development, and because of that also density of the ion channels mature because neurons needed to communicate more with other neurons. And this is actually the ion channels, and we know that the ions are important, so they go in and out of the membrane, and they create like they create basically electricity. So neuron firing is dependent on sodium and potassium, and deficits in ion channel have been also associated with ASD. And so basically what we can do with patch clamp is actually measure and by watching positive current flow in and out of the neurons, and we can quantify these. So when we quantify these actually at the in vitro 30, this is the outward potassium currents. What we see is that there is a significant difference between the control lines and the CHAMP1A, and also control lines in CHAMP1B, but kind of you can clearly see that CHAMP1A is very different compared with CHAMP1B. When we do the same for sodium, we have more or less a similar phenotype. We have like basically the CHAMP1A that is more severe compared with the CHAMP1B, and the control lines are here. But what happens in the in vitro 60 actually is very interesting. Like in the potassium, the outward currents, CHAMP1B actually pick up really well, while the CHAMP1A is still a huge deficit. And it's the same also for the sodium. So what we can actually summarize here is that there is a reduced density of sodium and potassium ion channels across development. CHAMP1A showed no increase in ion channel density over time, while CHAMP1B exhibit an increase of ion channel density between the 2 time points. It kind of pick up really well. So now we wanted to ask actually another question. Does CHAMP1 loss of function alter synaptic activity in vitro? So synapses are the junction between the neurons. They are really important to communication. They have, they communicate through neurotransmissions and so on, and you can actually quantify these. So for instance, this bar plot is showing actually synaptic activity across time. This is like basically what you should expect from a neurotypical line. But what we have here, you can see clearly like a difference. It is a day in vitro 30. You can clearly see a difference between CHAMP1A and CHAMP1B and the control lines. So when we quantify the percent of total neuron that have like actually synaptic activity, more or less you expect like more or less 80% of the neurons showing like a strong activity. And this is the control line. CHAMP1A actually have like a severe deficit. We have 25%, 48%, 50%, more or less at the in vitro 60, while again, CHAMP1B actually pick up really well at the in vitro 60, suggesting that we have two different phenotypes from two different actually mutations. We next also wanted to understand the neuronal structure of the neurons. So this is like a Scholl analysis. So basically you can quantify the number of dendrites, the distance from the soma and so on based on this kind of analysis. It's called Scholl analysis, okay? This is a reconstruction of the neurons at the in vitro 30 of a wild type, of a neurotypical. And you can see that the branches and so on, so you can quantify the distances and so on of these axons and dendrites. So this is like basically the number of intersections, in the Scholl analysis, and you can clearly see that it is a neurotypical. You can clearly see that the number increases over time, so the neurons become more complex. But what we have here is actually CHAMP1A and CHAMP1B. So at the in vitro 30, there is like a really severe reduction, also the in vitro 60 for CHAMP1A, but CHAMP1B actually pick up again. So suggesting that there is a delay maturation CHAMP1B, but kind of pick up really well by the in vitro 60, while the CHAMP1A stays like more or less affected. So in summary, there is a CHAMP1 loss of function seems to reduce actually the neuronal complexity. CHAMP1A showed deficit in the neuronal complexity, while CHAMP1B exhibited like a less severe phenotype. So now I'm talking about the neuronal connectivity using the MEA, so multi-electrode array. This is like a representation of the multi-electrode array. What we are doing in the lab, this is like still preliminary. We are collecting more batches of neurons. So what we have here is a plate from a multi-electrode array, and here there are all the electrodes, so we can actually cultivate the neurons here, and we can basically see how well they connect and how well they oscillate together. So what we can quantify is the activity, the firing rate, basically recapitulating actually the patch clamp. We can understand the synchronicity and the connectivity of the neurons, and also the oscillation. Are they like basically firing at the same time? So here I'm showing, again, this is very preliminary because we have only CHAMP1A and CHAMP1B right now. We are like doing like multiple batches. Here, oscillation, synchronicity, and firing rate. So again, what you can see here, especially for instance here, what we see is that the CHAMP1B is very different from CHAMP1A. The CHAMP1A kind of firing like across time do not perform really well. So basically they stay the same, while the firing rate actually increase over time for CHAMP1B. For the synchronicity is the same. We have like a better basically synchronicity for CHAMP1B, but not for CHAMP1A. And here the oscillation is the same. We have better oscillation for CHAMP1B, but not for CHAMP1A. This again like this is like a summary. CHAMP1A showed no increase of synchronicity and number of oscillation across time, so it's a really severe phenotype in these 2D neurons. And the CHAMP1B actually increases synchronicity and number of oscillation across time. And here, like a quick overview about genomics. We just got the data 2 days ago, so I just wrap up like a little some slides for you. So what we did was actually, we wanted to understand whether CHAMP1 loss of function altered the transcription landscape of developing neuron. And how we do that, we basically perform single nuclear RNA-seq data from the in vitro 30 neurons and the in vitro 60. Thank you, Valerio, for explaining the single cell, so I don't have to do any of this. So we basically, what we can do is, we collect the nuclei, we quantify basically gene expression, and we sequence. And here I'm showing the U map, or like basically the map of what we have. We have basically neuroprogenitors. Not all the cells become neurons. We have intermediate glutamatergic neurons. We have mature neurons. and we have like some other cell lines, cell types. Sorry. We tested like with multiple approaches. This is like another model, another method to understand whether these ones were really differentiated or not. So we can clearly see that these are cells that are differentiated. So these are postmitotic neurons. And here we see like more pluripotent cells like the neuroprogenitor. So we confirm basically that we have both a cell line neuroprogenitors, intermediate, and also differentiated neurons. They show like markers for for gene expression that are canonical for like these cell types, and also our genes like CHAMP1, POX-zeta, and so on are actually highly expressed in those lines. So we know that CHAMP1 is also expressed in postmitotic neurons. And here I'm showing like a big map. So as I mentioned before, this is like very preliminary. So we just got the data. So we compare CHAMP1A versus control for the multiple cell types that we have in our UMAP here in CHAMP1B, CHAMP1A, and CHAMP1B. Okay, the in vitro 60 and the in vitro 30. What you can clearly see, probably appreciate here, is that in the CHAMP1A we have like more changes. Check the bar plot in red. We have more changes both in the in vitro 30 and the in vitro 60 compared with CHAMP1B, suggesting that there are like more transcriptional changes in CHAMP1A. And if we do like an overlap between the genes that are differentially expressed in CHAMP1A compared with the CHAMP1B, and we want only to focus on the CHAMP1A because we see like more differences, what we found, for instance, if we focus on the neurons, we found that the genes associated with specifically with CHAMP1A are important for synaptic and axogenesis, suggesting that the changes in CHAMP1A are actually affecting the synapses and the morphology and the development of neurons in addition. So this is the summary. CHAMP1A loss of function affect the transcriptome in human brain development. I think you appreciate that. You appreciated that. The CHAMP1A actually show a higher impact in the transcriptome landscape of developing neurons compared with CHAMP1B, and what we found is that genes associated with synaptic function, neuronal development are actually affected, kind of correlate with the electrophysiological deficit that we found in CHAMP1A compared with CHAMP1B. And this is just like a overview and the summary, and also like a working in progress, what we are doing in the lab right now. Basically, I think you appreciate that the CHAMP1 mutation halted neuronal firing, ion channel density, and synaptic activity. But what we found very interesting is that the two different mutations lead to two different phenotypes that are actually correlated with the phenotype of the kid. The CHAMP1A, the arginine 497, displays a general delay in maturation and pronounced electrophysiological deficits, showed a reduced synchronicity, absence of oscillatory activity, and exhibits transcriptomic alteration correlated with electrophysiological impairments. While the CHAMP1B, why it doesn't work now? The CHAMP1B displays a mild delay in maturation with moderate electrophysiological deficits, shows increased synchronicity, oscillatory activity. But as I mentioned before, we're still working. It is like a working in progress. We are like expanding the experiments, and exhibits limited transcriptomic changes compared with the CHAMP1A. It's kind of like, reflecting the fact that basically we have two different phenotypes that kind of correlate with the phenotype of the kid affected by these mutations. So as I mentioned before, we have a working in progress. So what we are doing is expanding further the electrophysiological experiments. We have like multiple lines right now, like again, like a knockout, a full knockout, and another neurotypical, just to improve a little bit like the data. Daily, my postdoc is analyzing the data as we speak. We are waiting for the single-cell genomics for forebrain organoids. We map basically organoids from the in vitro 30 to the in vitro 120. Zach did the majority of the experiments for this, basically. And we are waiting for the genomics, so we will have also this one to back up what we see like in the 2D neurons. And also we are like working with epigenetic signatures during brain development. This is like the project of Zach, but we also collaborate with Kozo Tanaka on this. So with that, I would like to thank my lab, my amazing lab, especially Daly, Christina, and Zach, who is here. Daly did like the majority of the work on the electrophysiology. She's an amazing electrophysiologist. Christina did like the majority of the work with the stem cells and so on, and Zach is doing like an amazing work with this translational part, but also the epigenomic. I would like also to thank Kozo. He's an amazing collaborator. We work a lot together, especially on the epigenomic side. And of course, I would like to thank the CHAMP1 Foundation and also the kid. Thank you so much.
Thank you, Stefano. Other questions for Dr. Berto? It's okay? Okay.
You got lucky.
Oh, Marcella. Pensavi di cavartela.
Thank you, Stefano. So your preliminary message is that maybe a mutation in the end of the protein can be milder?
Yeah, we don't. Yeah, it's an open question, right? We don't know. It's an open question. We don't know. So our idea that this is like why we are doing like also the following up, you know, with the with the epigenomics is that the function of CHAMP1 and interacting also with HP1 alpha might be like really like one of the models to follow, especially in post-mitotic neurons. HP1 alpha is important for heterochromatin. You condensate the heterochromatin. If CHAMP1 do not bind anymore with the HP1 alpha, might lose the chromatin structure and so on, and therefore like gene expression might be like completely random, and therefore affecting you know, the gene regulatory mechanism. So it's a good question because the CHAMP1B should basically translocate into the nuclei because it still have like the translocator motif and potentially have like some function binding again the HP1 alpha. So probably it's milder for that. We don't know that. So that is a good question. So.